What looks like a confusing traffic light is just a very tiny DNA microarray. DNA microarrays, also known as DNA chips, are tools which are used for gene expression analysis and for genotyping. Please note that DNA chips come in different flavors. Before we reveal the principle behind one of those DNA microarrays, I encourage you to subscribe to my channel. In the lower right corner, you can find the subscribe button. The first step for microarray analysis is the sample preparation. In this example, we have samples 1 and 2. Sample 1 might be tissue from a healthy cell, and sample 2 could be cells from tumor tissue. The RNA is extracted from both samples and then transcribed into cDNA using reverse transcription. During this step, the cDNA is fluorescently labeled. cDNA from the healthy sample 1 in green and cDNA from the malignant sample 2 in red. Sample preparation is complete. Taking a closer look at the microarray plate, a chip has many spots. Looking more closely into one of them, each spot is coated with multiple identical probes. These are short oligonucleotides that cover the sequence of a specific gene. Now we can observe the spot of gene X when we add our samples. In this example, molecules from sample 1 hybridize with the probes of that spot. Sample 2, however, does not contain complementary cDNA molecules and is washed away. The spot we were looking at appears green after laser scanning, which tells us that gene X is expressed in sample 1, but not in sample 2. Examining a different spot on the microarray containing different oligonucleotides against gene Y, we might observe the opposite scenario, with only sample 2 containing complementary cDNA, but not sample 1. In this case, the spot of gene Y appears red after scanning because of the specific fluorescent label. As a conclusion, gene Y is not expressed in sample 1, but in sample 2. The third possible scenario is that both samples contain molecules that may hybridize with a gene's probe, as shown here for a hypothetical gene Z. After laser scanning, the spot of gene Z looks yellow because both fluorescently labeled samples are present after washing. This result tells us gene Z is expressed in both samples, in the healthy tissue and also in the tumor tissue. In reality, the cDNA sample is added to the whole plate in one step. During microarray chip analysis, a laser excites the fluorescent dye and the emission levels are measured by a detector. Raw data can then be analyzed using bioinformatics. Check out the latest video here or take a look into next-gen sequencing. And make sure to like this video and subscribe to this channel to get notified immediately when the next fresh video is uploaded. Thanks for watching.